Good day, everyone. Today we will be talking about bacterial identification in the clinical bacteriology laboratory. Now, the most important tool you will use in identifying your bacteria is microscopy. Given that bacteria and other pathogenic microorganisms are very small in size, you will not immediately see them using your naked eye if they are present in your sample or not. So, you need to use the microscope, which is one of the best tools you can use to view, identify, determine, and differentiate these microorganisms. In the microbiology laboratory, you will encounter a variety of microscopes. However, the most commonly encountered one is the bright field microscope, otherwise known as the light microscope. This microscope uses light rays to produce a dark image against a bright background. However, this microscope also has the disadvantage because bacterial cells are not clearly seen unless stained or suspended in non-aqueous solutions that enhance refractive index. This is because your bacteria have a similar refractive index to that of water, which means that if you look at them unstained under the microscope, they will seem transparent or semi-transparent. So you need to use stains or similar materials to change its refractive index. Next, we have the phase contrast microscope. This microscope uses a special optic system which converts phase shifts in the light passing through a transparent specimen to brightness changes in the image. So when we say phase shifts, light passing through the specimen is sometimes blocked by thicker parts of the specimen, and this causes the light to shift. So in the top part of this slide, you can see the principle of the phase contrast microscope. As the light passes through the specimen, it produces both unretarded and retarded light rays. And this passes through the objective and into the phase plate, which separates the two, and this allows a difference between the types of light rays to be observed in the image plane or through the viewer's eye. So the difference can be seen in the lower part of the slide. In letter A, these are brain cells that are seen under the bright field microscope. And you can notice that there is very little contrast in these cells and they're almost transparent. You can hardly see any internal structures at all. However, using a phase contrast microscope in letter B, you can suddenly see the internal structures of the cells as well as the external structures. Next, we have the dark field microscope. This uses a special condenser to block direct illumination and directs light at an angle so that no incident light reaches the objective. Right? So you can see that on the left side of the figure, you have the special condenser which has a blockage in the middle, and this allows only light to enter at a certain angle. And when this light reaches the sample, it is redirected away from the objective. So this is what we call a hollow cone of light. In contrast, the bright field microscope allows all light to enter the condenser and into the sample and then into the objective. So using a dark field microscope, you will see samples that appear bright against a dark background. Next, let's go into electron microscopes. There are two types. The first is the transmission electron microscope. And here, a beam of electrons is transmitted through an ultra-thin section of a specimen to form an image which can demonstrate bacterial or cellular ultrastructure. Right? So here you can see the principle of this type of microscope. So the electrons pass through the sample and then into your lenses. Right? And this is how you visualize the insides or the microstructure of your cells. Now, we measure how close or how small the objects under the microscope can be seen in what is known as resolving power. So that is the ability of the microscope to distinguish two close images as being separate. Now, your transmission electron microscope, or TEM, has a resolving power of 0.001 micrometers, which is about 200 times greater than the bright field microscope. Then we have the scanning electron microscope. And in contrast with the transmission electron microscope, which is used to see the insides of the cell, the scanning electron microscope is usually used to see the outside or the external structures of the cell. Alright, so a primary electron beam is bombarded onto the surface of the specimen, causing the emissions of secondary electrons, which then produces a tridimensional or 3D image. So one thing to note here is that your sample has to be coated with a substance that does not allow your electron beams to pass through. And this allows the electrons to be deflected and then subsequently detected by a secondary detector. And this gives us our 3D structure of our cell or bacteria. Now this gives us a resolving power that is actually weaker than the transmission electron microscope at only 0.005 micrometers. However, if we compare this to our bright field microscope, this is actually still much greater. Okay, so before you're able to look at your bacteria under the microscope, you first have to place them onto glass slides. And the process of doing this is called bacterial smear preparation. You can do this on a variety of samples. Now, if you're taking a sample from an infected body part or material, this is what is known as a primary smear. But if you are taking bacteria from a solid medium or a liquid medium that has already been inoculated previously, this is what we call a secondary smear. Now for the process of creating a smear using a solid medium. The first thing you need to do is soak a clean glass slide in denatured alcohol and set the slide aflame by passing it over an alcohol lamp. And this causes the alcohol on the slide to burst into flame, essentially sterilizing the slide. Then you need to cool the slide down until you're able to touch it and then label it properly. After that, place a drop of sterile water on the glass slide. Next, you obtain a small colony of bacterial growth by lightly touching the surface of a colony with your inoculating loop. However, if you are dealing with very tiny or pinpoint colonies, you can even take the entire colony. Alright, and then you emulsify the obtained bacterial colony within the drop of sterile water, and you spread that across the slide to an area of about 1 centimeter squared. If you need to, you can spread the bacteria in a wider area so that you just have to make sure that the bacteria are uniformly distributed so that you are able to see the individual cells of bacteria. Alright, after that, you air dry the smear and heat fix it by passing the slide rapidly over an open flame around 2 to 3 times. So each time you do this, you will notice that steam accumulates on your slide each time you pass it through the air, and this is actually your excess water being evaporated, right? And you know that you are done heat fixing once you pass the slide through the flame and no more steam forms. Okay, don't forget to allow your slide to cool before staining. Now, if you are using liquid medium, you still sterilize the slide the same way, and then, observing aseptic technique, obtain a loop full of organisms from your liquid medium, like a broth, and place it at the center of the slide. spreading to about 1 square centimeter. Right? And then again, you air dry the smear and heat fix it by passing it through the flame. And then you allow it to cool before staining. Now, if you are using a swab, it's actually very simple. Again, you begin by sterilizing your slide by lighting it on fire. And then you take your swab and you roll the swab back and forth over an area of your slide to deposit a thin layer of sample onto your slide. And then you air dry the smear. Alright. Note that smears should not be prepared from a swab after it has been placed or used to inoculate culture media. And the swab should never be rubbed back and forth across the slide. Rather, you should roll your swab on the surface of your glass slide. Now, if you are dealing with a thick liquid or a semi-solid, such as stool or sputum, again, you need to sterilize your slide and then immerse a sterile swab into your specimen for several seconds and allow your swab to absorb the specimen. After, prepare a thin spread of material on the glass slide by rolling your swab. And once again, air dry the smear before heat fixing and staining it. If you are using a thin liquid, such as urine, CSF or cerebrospinal fluid, and different transudates, you again need to sterilize your slide and then make a circle on the reverse side of your slide. This is to maintain a boundary for your smear. And then you drop around 50 microliters onto the pre-marked area. Now, you do not spread this. You don't spread this liquid. Rather, you allow it to dry within the pre-marked area. And after that, you can heat fix and proceed with staining. Alright, now let's talk about bacterial staining. Stains impart color or fluorescence, in case you are using a fluorescent stain, onto the bacterial cell wall, which further visualize the organism under the microscope and help in the classification according to their staining characteristics, shapes, and groupings. Now we have different types of stains. The first is the simple stain, which only uses one dye or one color to stain the entire structure or the whole cell. Right? Then we have your differential stain, which uses one dye in the staining process to divide bacteria into two groups. Alright, so we have an example of a differential stain on the left side of this slide. Right, so you have one stain for one type of bacteria, and you have another stain which stains another type of bacteria. So you can differentiate them based on which dye has stained them. Then again, you have your negative stain. These are usually used to study the morphological shape, size, and arrangement of bacterial cells that are difficult to stain or those that are too delicate to be heat fixed. And then finally, you have your special stains, which are used to emphasize certain structures or features of an organism. For example, you can have special stains that only stain the flagella or the locomotive organelle of your bacteria. For your differential stain, there are two main ones being used. That's your gram stain and then your acid-fast stain. Alright, let's look at some staining procedures. The first one we have is simple staining using only one dye, and here we're using crystal violet. Alright, to do this, you simply need to flood your dried and fixed smear with crystal violet for about 30 to 45 seconds or whatever time that you have established, and then using an indirect stream, rinse the excess stain with tap water. Right? And then you can gently dry the slide either by blot drying it with a paper towel or by air drying. However, we recommend that you air dry your slides because this prevents you from touching the smear and possibly disrupting it. So it's better to just leave it at room temperature to air dry. Then we have our gram staining. This is an example of a differential stain, meaning one type of stain is used to color certain bacteria and another type of stain is used to color the remaining type of bacteria. Right, for the procedure, flood dried and fixed smears with crystal violet for 1 minute. And after that, you rinse the excess stain with an indirect stream of tap water. Make sure again that it is really an indirect stream because if you hit your smear directly with your water, this can cause some cells to be washed off. All right, and then you flood the slide with grams iodine for 1 minute. This is what we call the mordant, and this allows the primary stain. In this case, when we say primary stain, that is the first stain that we apply. This allows the primary stain to adhere better on the surface of our cells. All right, and after that, you rinse again using tap water. After that, you flood with 95% ethyl alcohol. This serves as your decolorizer. And when we say decolorizer, this removes stains from cells which did not adhere to those stains. So for example, if you have a certain type of cell, usually these are what we call gram-negative bacteria that have a very thin cell wall. These bacteria are not able to trap your primary stain. So when you add your decolorizer, this washes out the primary stain, right? And after you add your decolorizer, again, rinse it immediately with tap water. After that, you add your secondary stain for 30 seconds. And our secondary stain in this case is safranin. So the purpose of the secondary stain is to stain all the other cells whose primary stain have been washed out. Okay, so the secondary stain then takes the place of the primary stain. Then you blot dry or air dry your smear before viewing it under the microscope. Next, we have another differential staining method known as the acid-fast staining. So acid-fast staining has two subcategories. The first one we will be discussing is the Zeal-Nielsen technique or the hot method. Right? This is usually used for differentiating mycobacteria. You see, mycobacteria have special components in their cell wall, such as your mycolic acid and a sort of waxy complex. All right, so usually these components do not allow your dye to penetrate the cell wall of your bacteria, of your mycobacteria rather. All right, so in order to stain these cells, you have to follow a specific set of instructions. So the first thing you need to do is to flood the dried and fixed smear with carbofuchsine, that is your primary stain, and then you heat the slide to almost boiling. So you will notice that you've heated your slide enough when you notice there is steam rising from your slide. Okay, you do this by passing the flame of an alcohol lamp underneath the glass slide. Okay, this allows or this causes those compounds which we talked about earlier, the mycolic acid, this causes them to become penetrable by your primary stain, right? And then you cool down your slide by gently washing it with tap water. Make sure to shake off the excess water. After that, you flood the slide with an acid alcohol decolorizer for 30 seconds only, right? So make sure that you don't go past 30 seconds. And this decolorizes all other bacterial cells except your mycobacteria. Right? And that's because the complex, which we made penetrable earlier by heating, now it is cooled down, and the stain that is already inside the cell wall is not able to escape because, again, the complex of mycolic acid and wax is impenetrable to your decolorizer. All right. Then after you put your decolorizer, again, rinse well with tap water and counterstain with methylene blue for 30 to 50 seconds. So your methylene blue will stain all other parts of your slide except for your acid-fast bacteria, which were already stained with your carbofuchsine. Again, you rinse and then air dry before you view these slides under the microscope. So you have an example of your acid-fast staining at the bottom of the slide. You can see that acid-fast positive bacteria are red because our carbofuchsine is a reddish stain. And if it is an acid-fast negative bacteria, they will stain blue. Also, the rest of the slide, any cellular components of the slide will also stain blue. All right. It's also important to note that when you are heating your slide using the Ziehl-Nielsen technique, do not let the stain dry out. Next, let's take a look at the next acid-fast staining method known as your Kinyoun method. So in contrast to the Ziehl-Nielsen method, the Kinyoun method does not use any heating to allow the primary dye to enter our mycobacteria. Rather, it uses other compounds. In this case, it uses phenol, which is added to your primary stain to allow better penetration. All right, so to do this, you first need to flood the dried and fixed smear with your Kinyoun's carbofuchsine for 5 minutes. Again, the Kinyoun's carbofuchsine is a combination of basic fuchsine and phenol. After, you rinse with distilled water and decolorize with acid alcohol for 3 minutes until faint pink, or you see that your slide is faint pink, and then you rinse again with tap water. After you do that, you can counterstain by flooding with methylene blue for 30 seconds. And then you rinse and dry your slide before viewing it under the microscope. So the appearance of acid-fast bacteria using the Kinyoun method is still the same as that of the Ziehl-Nielsen technique, in which acid-fast bacteria are stained red, while non-acid-fast bacteria are stained blue, as well as the background. Next, let's take a look at our different dyes that we use for staining. So there are two classes of ionizable dyes. You have your basic dyes. These are dyes with positively charged groups. Otherwise, these dyes are cationic, and these dyes bind to the negatively charged molecules of your cells, such as your nucleic acid and many other proteins. Included in these are the bacterial surfaces or the bacterial cell walls, which are negatively charged. And so basic dyes are usually used as the stains of bacteria in the microbiology section. Right? Now, we have acidic dyes as well, and these are anionic dyes, or they have negatively charged groups that bind to positively charged cell structures. Examples of these are eosine, rose bengal, and acid fuchsine. Let's now take a closer look at gram staining. This was developed by the stain's namesake, Hans Christian Gram, to divide bacteria into two large groups. You have the positive, the gram-positive, those that take up the basic dye, which is crystal violet. Usually gram-positive bacteria have a thick peptidoglycan layer and numerous teichoic acid cross-linkages which contribute to the bacteria's ability to resist alcohol decolorization. So here you can see the gram-positive cell wall. It is a very thick peptidoglycan wall, so any stain that is able to penetrate this cell wall is not able to escape owing to the thick peptidoglycan layer. Then you have your gram-negative bacteria, and these are bacteria that allow crystal violet dye to be washed out easily, and this is because they have a thin peptidoglycan layer compared to your gram-positive bacteria. All right. They also have a thick external coat of lipopolysaccharides and proteins. Right. So you can see the gram-negative cell wall on the right side of the slide. Notice that the peptidoglycan layer is much smaller compared to the gram-positive bacteria. Alright, what are the general rules when looking at our gram staining results? So these are rules that help us identify our bacteria quickly based on the gram stain alone. The first one is that all cocci are gram-positive except Neisseria, Veillonella, and Moraxella. So previously Moraxella was known as Branhamella. Right? So if you see a gram-negative cocci, most likely it is one of these three genuses, and you can immediately report that to your clinician to help them diagnose and give treatment to your patient faster. Then you can also note that all bacilli are gram-negative except the following: Mycobacteria, Corynebacteria, Clostridia, Bacillus, Listeria, Erysipelotrix, Lactobacillus, Actinomyces, Eubacterium, Propionibacterium, and Bifidobacterium. Of course, there are other bacteria in the environment that are also gram-positive. However, these ones here are the ones that are usually found or usually encountered in human samples. Also note that all spirochetes are gram-negative bacteria. Spirochetes are bacteria with a spiral shape. Now, when we are doing our gram staining, you have to take note that there are some factors that can cause an inconsistent gram stain reaction. So, for example, gram-positive bacteria can sometimes stain as gram-negative in the following cases. The first one is removal of magnesium RNA by precipitation with bisalts. Next, you have autolysis, aging, and temperature of incubation. So this results to a loss of cell wall integrity, and because the cell wall integrity is decreased, it cannot hold the primary stain and therefore is easily decolorized. So it's always good practice that when you are staining your cells, make sure you are using a fresh culture, right? So at least 18 to 24-hour culture for your gram staining, right? And then if you also used an acidic solution of grams iodine, this can cause your basic stain to not bind as well to your cell wall, right? And of course, if you over decolorize your cell wall, this can cause excess removal of your primary stain. Next, we have reasons why gram-negative bacteria sometimes become gram-positive. Right, the first one is an incomplete decolorization. So if you did not decolorize your smear well enough, there might be some leftover primary stain in your cells, and this causes, or this does not allow our secondary stain to stain our gram-negative bacteria. Then if you also have a thick smear preparation, this can cause a purple appearance to your cells, especially if these cells are found or these cells are covered by other bacteria. Then you have if your iodine is not thoroughly washed off, and if you also have a crystal violet that is too strong or has a very high concentration, or is left too long on the smear, or even if you have crystal violet sediments in your stain bottle. So this is why we have to filter our crystal violet before we stain or before we use it to stain our cells. The next thing we'll be discussing is acid-fast staining, right? We classify bacteria as acid-fast when they have long chains of fatty acids or mycolic acids in the cell wall. This gives them resistance to acid alcohol decolorization, thus retaining the basic dye, which is in this case is carbofuchsine. So a good example again of acid-fast bacteria are your mycobacteria, the causative agent of your tuberculosis and your leprosy, right? So leprosy is caused by Mycobacterium leprae, and tuberculosis is caused by Mycobacterium tuberculosis, right? So mycobacteria do not readily stain, but with the application of heat or increasing concentration of basic fuchsine and phenol, the stain is allowed to enter the cell wall. So you can see here that your mycobacteria have this layer of mycolic acids and proteins on top of the cell wall, and this need to be disrupted using your heat or phenol to allow your primary stain to color the cell wall. All right, so mycobacterium, once they are stained, they cannot be decolorized even with acids. So they are called acid fast, meaning they hold fast in the presence of acid. All right, so non-acid fast microorganisms cannot resist acid alcohol decolorization and will readily stain with a counter stain. So, what are some ways to facilitate acid fast staining of your mycobacteria? So, as we discussed earlier, either you can steam it, that is your Ziehl-Neelsen technique, or you can increase the concentration of basic fuchsine and phenol, that is your Kinyon method. You can also have prolonged contact of the stain with your bacteria, and you can add agents like tergitol to the stain solution. So these are what we call wetting agents, which decrease the surface tension of a stain for better permeability into the cell wall. Okay, here we have some other methods of acid-fast staining. The first one is Pappenheim's method. This differentiates M. smegmatis from M. tuberculosis. Right? Using this method, Mycobacterium smegmatis is decolorized by a mixture of rosolic acid and alcohol, making it blue. However, Mycobacterium tuberculosis remains acid fast and therefore it's still red. Then you have your Bog-Martens method, and this is used to differentiate Mycobacterium tuberculosis with Mycobacterium leprae. Alright, so Mycobacterium leprae is easily stained by dilute alcoholic fuchsine, and this is what we use as your primary stain. Meanwhile, your Mycobacterium tuberculosis will still appear as blue. Next, we will discuss another method of identifying our bacteria based on their motility or their ability to move. Okay, so many bacilli and spirilla are motile by the use of flagellum. Now, because of their thinness, the flagellum's thinness, they cannot be directly observed under the bright field microscope, and thus you need to stain them with special stains or use other techniques to observe the flagellum. Okay, so we can classify the presence of flagella using these terms. When we say atrichous, this means that the bacteria has no flagella. Now, when you say monotrichous, there is a flagella at one end only of the bacteria, a single flagella. Then we have amphitrichous, where your bacteria has flagella on both ends. Then you have Lophotrichous bacteria, which have a clump of flagella at either one end, or even sometimes you can find this on both ends. But you should remember that Lophotrichous refers to a bacteria that has a clump of flagella or multiple flagella on one end. Then you have your Peritrichous bacteria, and these are bacteria with multiple or numerous flagella all over its body or its um, cell surface. So let's look at some methods to detect motility. The first one is through the hanging drop method. So this method is used for the continuous observation of motility or for monitoring the growth of individual cells in a microculture. So to do this, you simply have your cover slip and you place um, a bit of clay on each corner of the cover slip. This will be used to raise the cover slip later on. And then you add 2 loopfuls of organisms on the center of the cover slip. Alright? Then you use a special slide with a depression in the middle, right? Similar to the one you see in number 3. So the depression side is pressed against the cover slip. Aside from clay, by the way, you can use Vaseline to allow your cover slip to stick to your slide, right? And then once you do that, you invert the slide so that the cover slip is facing up. So now you have a drop of liquid that is suspended on the cover slip. All right, so any bacteria in that drop of liquid, you will be able to see them if they are moving. Okay, and you should not confuse the movement of bacteria using the hanging drop method with Brownian motion. So to differentiate them, you have to look at your bacteria and you observe the type of movement they are showing. Okay, so if it's like an independent movement or if they're moving at different directions, the different bacteria, then we can consider that as true motility. But if all the bacteria are moving at in one direction and it seems like they have no choice or like they are all moving there out or without any change of direction, then we can assume that this is caused by Brownian motion. Also, it's an important thing to note that you should use very young cultures when you are doing your hanging drop method. So if you're using a broth culture, they should be at least 4 to 6 hours old. And if you're using colonies from agar plates, these colonies should be 24 to 48 hours old only. Next, we have our flagellar staining. So as I mentioned earlier, flagella are very, very thin, so it's hard to visualize them using our bright field microscope. So to be able to see them, we have to thicken them using stains. The first one is Gray's flagellar stain, and it uses a mordant to swell the flagella for better staining. In contrast, Clark's flagellar stain is uses tannic acid as a mordant. Then we have another method which we discussed in a previous video. So you can demonstrate motility by using growth in a semi-solid medium. All right. All you need to do is touch the top of a well-isolated colony using an inoculating needle and inoculate it to an SIM medium or a SIM indole motility medium by stabbing through the center at around 2/3 of the tube's depth. Then you incubate your SIM medium for 18 to 24 hours at 35 to 37 degrees Celsius. Now, if your bacteria is motile, you should notice a diffuse growth from the line of the inoculum, meaning your bacteria has moved away from the line of inoculum towards the rest of the tube. So here you have some examples. This is your gram negative, your motility negative bacteria on the left side. You can see that the bacteria are confined in the stabbing areas of your tube. Meanwhile, for the motility positive bacteria, you can notice that the tube, the whole tube in this first example is cloudy, so meaning the bacteria have migrated throughout the tube. And here in the second example, you see that the area surrounding your stabbing line is cloudy, meaning that bacteria have emerged from the stabbing line into the surrounding area. All right, and don't forget that when we say semi-solid medium, these are usually agar with a concentration of 0.5% or less. Okay, then we also have other ways to demonstrate motility. You can do serologic tests. These are tests that use antibodies to detect the presence of the flagella. Then you can use the fluorescent antibody technique, or FAT, and this uses antibodies or antibodies for the flagella with fluorescent dyes attached. Also, if you notice a swarming phenomenon on your culture plates, meaning your bacteria have swarmed or moved throughout the entire plate, this is a characteristic of Proteus, and this is also a way for us to indicate the motility of the bacteria. And finally, if you use dark field microscopy, you can also be able to demonstrate motility. Okay, so that ends our relatively long lecture for today. If you would like to read more about the things discussed here, you can check out these references. And for more videos on clinical laboratory science, please consider subscribing to this channel. Thank you very much for listening.